Good morning, respected all. Today we have a very important topic to discuss. That is multi-drug resistant bacterial infections, whether it is an emerging challenge or not. You all know that antibiotic per se, the discovery of which and the introduction into the clinical use has been one of the greatest medical breakthrough of the 20th century. It has saved millions of lives, as we know. It not only helped us in combating most of the infections, but also it let us do all the medical procedures and complicated surgeries, transplantations, chemotherapies, etc., etc. So over the last 100 years of its discovery, it has extended the average human life by almost 25 years. But during the reception speech of his Nobel Prize for discovering the antibiotic, Alexander Fleming himself predicted rightly, even 80 years ago, that the mankind is going to mishandle it, mismanage it, misuse it, abuse it, eventually lead on to drug resistance. Tom Frieden, who was the former director of CDC, said, if you use antibiotic when not needed, we may not have them when they are most needed. So currently, as per the CDC calculations, the AMR or the antimicrobial resistance costing around 1.27 million death worldwide. It is actually just like COVID-19, we are into a slow-moving pandemic. It's called AMR or antimicrobial resistance. Not only to bacteria, it could be virus, fungus, parasites, etc. And WHO has already listed AMR into one of the 10 threat to global health in 2019 itself. So this slide shows the global burden of the drug resistance infection worldwide. We can see that almost all the part of the world has affected, especially in the African region and South Asian areas. We can see that mostly the three the infectious process mainly in the lower respiratory tract, bloodstream infection, and intra-abdominal sepsis account for most of the death attributable or associated with the multi-drug resistance organism worldwide. Most common organism being E. coli, Staphylococcus aureus, Klebsiella pneumonia, Streptococcus pneumonia, and Acinetobacter baumannii. So mostly Staph aureus, Enterobacteriaceae, gram-negative bacteriaceae, and also Pseudomonas, meant tuberculosis. Our Arabian Peninsula is not so different. We can see that there has been multiple reports in this region, mainly from Saudi Arabia and Qatar, and also from Bahrain. We have reported Acinetobacter baumannii and E. coli, which are resistant to multiple organisms. Not only that, we can also see that as for the last 14 years, the trend of mortality is on the rise in this region. Mostly in Saudi Arabia, maybe because of the reporting bias and the size of population. And most of the organism in our region being a mixed organism, multiple MDR pathogens are on the first place. As far as the mortality is considered, followed closely by Acinetobacter and Mycobacterium tuberculosis, among other gram-negative Enterobacteriaceae. We can see that the mortality rate has been close to 20% if we have infection with MDR bacteria. So it's not only the healthcare or the healthcare system which is a culprit of making all these organisms resistant to multiple drugs. We can see that humans are not the only one, but antibiotics are being pumped and used in other field like agriculture, livestock, the fisheries. For the farm animals and birds are being treated with antibiotics. So in them also, the emergence of bacteria and the drug resistance genes can develop. And if these genes get contaminate the environment, the drinking water, and because of the poor sanitation and the sewage disposal, it can also affect the human being ultimately. So this is the whole process of how the MDR bacteria is and the gene is produced and stored in the environment. So it's not only the hospital or the healthcare facility only, but also it's a large scale problem and production of and exchange and spread of antibody resistance genes and bacteria. WHO has enlisted drivers of AMR. The first and foremost, of course, is a misuse and overuse of antibiotic, but also lack of access to clean water, water sanitation and hygiene wash, poor prevention and control of infection and disease in the healthcare facilities and farms, poor access to quality of affordable medicines, vaccine and diagnosis, lack of awareness and knowledge, and lack of enforcement of legislation. And it has enlisted many organisms into the priority wise and for the research and development of antibiotic against, and the critical ones are the Acinetobacter baumannii, which are Carbapenem-Resistant called CRAB, then Pseudomonas aeruginosa Carbapenem-Resistant, and also Enterobacteriaceae or CRE, Carbapenem-Resistant Enterobacteriaceae. So these three are the priority or priority one critical organism against which there has to be there is a need of immediate research and development of antimicrobials. But unfortunately, only three of newer molecules has been directed against this critical organism. Not more than that. Even though there are 17, only 17 means at least one of the WHO innovation criteria. Only 3 of them are actually against these critical organisms. So why there is a less investment in the research and development of newer molecule is because of course, high expense of research and growth of new molecule, but the low return due to the evolving antimicrobial resistance. The moment we have a new introduction of antibiotic, there'll be resistance in the immediate future. So only 5 to 20% of pharmaceuticals firms are now active compared to back in 1980 in the development of newer molecules and antibiotics. So we are into a post-antibiotic era. We are avoid of any or there is scarcity of newer antibiotics and molecule for these evolving organisms. So as per the WHO, the MDR or multi-drug resistance is considered if the organism is resistant to at least one agent in more than 3 antimicrobial categories. XDR is resistance to more than or equal to one agent in all but 2 categories, and pan resistance means resistance to all antimicrobial categories. So why bacteria develop, especially gram-negative bacteria develop resistance is basically 4 mechanisms. The efflux pump, which will efflux or pump out all the antibiotic that has reached inside, to pump out all the antibiotic outside. There are limiting the uptake of the drug into the bacterial cell. There is inactivation of the drug because of some enzymes and other molecules, and also modification of the target drug target, which will enable the antimicrobial ineffective. And some gram-negative bacteria produces all 4 mechanisms as well. So initially there was a wild type bacteria before the introduction of antimicrobial. Then we invented penicillin, but the bacteria was very smart enough to produce beta-lactamase like TEM1, 2, and SHV1. Then we developed in 1980s beta-lactamase inhibitors, which combined to beta-lactam, and also we produced cephalosporins like cefotaxime and ceftriaxone in 1980s. But soon after, the bacteria produced AMP-C and ESBL gene, which will enable these antibacterial also ineffective. And ultimately, we developed carbapenems like meropenem in 1990s, but we have carbapenemase now. So the moment we develop one antibiotic like penicillin 1943, then 1960 there was penicillin resistant pneumococcus. So the moment we develop newer antibiotic, there has been a development of resistance in the bacteria as well. So coming to the the carbapenemase enzyme classification, basically it is classified into 3: class A, B, and class D. Class A is the most prevalent one, and the most important one is the KPC or Klebsiella pneumoniae carbapenemase enzyme. Class B is called metallo-beta-lactamase, which contains zinc ion in this molecule, which is very capable of horizontal transfer from the bacterial population. So it can make most of the beta-lactam antibiotic ineffective except for the monobactam. The examples are NDM or New Delhi metalactamase 1. And class D is mainly OXA48 type. So what is the definition of CRE or Carbapenem-Resistant Enterobacteriaceae? If the Bacteriaceae or Enterobacteriaceae are resistant to at least one of the carbapenem or producing a carbapenemase enzyme is called CRE or Carbapenemase resistant Enterobacteriaceae. In CRE, it has two type of mechanism. One, as we already discussed, there are four different mechanisms like efflux pumps and porine mutation, etc. And it can produce carbapenemase enzyme, which could be any of the any of the three categories that we have mentioned already. It could be AMR A, B, or D. And most of the, as already mentioned, after the CDC data, the most of it being the KPC, almost 86% of the time, it is KPC gene and the enzyme. So when to suspect carbapenemase? For the microbial, we know that the MIs or minimum inhibitory concentration to imipenem and meropenem are above the EUCAST breakpoint. So if you find the MIC during the subculturing of the organism, if the MIC is beyond the breakpoint you put forward by the CLSI or EUCAST, then we call it or we suspect carbapenemase resistance. Then the next step should be whether it is a carbapenemase enzyme producing resistance or the other four mechanism producing resistance. For that, we have different microbiological tests like phenotypic test, or nowadays this molecular testing is available like multiplex PCR testing to detect the genes like APC gene or NDM1 gene like that, because it has got therapeutic implication. Suppose the organism is not producing the enzyme, but having the other mechanism like efflux pump, etc., then still we can use meropenem in a higher dose with an extended infusion. Like this slide, you can see that extended infusion meropenem 2 gram 8th hourly infused over 3 hours can actually overcome all the resistance pattern if it is sensitive to meropenem and is not producing the carbapenemase enzyme. If it is producing the carbapenemase enzyme, then we may have to go for other antibiotics. And nowadays we have a novel broad spectrum beta-lactamase molecule like avibactam, relebactam, vibrobactam, etc. But there is no intrinsic antibacterial activity for this molecule, unlike sulbactam. It does not have a antibacterial activity of its own. It has to be combined with other antibiotics like ceftazidime, avibactam combination, meropenem, vibrobactam, imipenem, cilastatin, relebactam, and ceftalozine, tazobactam combination. But it will not inhibit the class B metallo carbapenemase, class B, AMR class B. And this combination, the ceftazidime, avibactam, meropenem, and amibactam can affect AMR A and AMR C, except Acinetobacter and AMR B or metallobeta-lactamase producing organisms. So we put the antibiotic into the previous slide. We can see that for class A, if we find out the by molecular testing or phenotype testing the nature of the carbapenemase enzyme, we can use ceftazidime, avibactam, meropenem, vibrobactam, or imipenem, cilastatin, relebactam. If it is class A, it is OXA type, we can give ceftazidime, avibactam. But we still not have anything for the class B metallobeta-lactamase. For that, we have to combine ceftazidime, avibactam with aztreonam, because that monobactams are still effective in those enzymes. So we have to combine ceftazidime, avibactam with aztreonam, or there is a newer molecule called ceftriaxone, which can also be used in this situation where we have metallobeta-lactamase producing CRE infection. We can see that if you are using monotherapy with the ceftazidime, avibactam, or aztreonam monotherapy for metallobeta-lactamase, there has been no inhibition of the growth. But if we combine these two, then we can have a good sensitivity pattern. We can see that the growth has diminished in with the combination of antibiotic. And this ceftriaxone is a newly developed US FDA approved first in its class antibiotic, which is a siderophore cephalosporin with a novel mechanism of action, which will not only uses this porine channel, but also it uses the ion transport system. With the ion, it can enter into the cell wall of the bacteria, and it can affect it can have this antimicrobial effect. So it is active against all major carbapenemase that we have discussed already. Now coming to the next organism, the carbapenem-resistant sustainable bacterial or the crab infection, which is very difficult to treat because first of all, there is confusion between the colonizer or it is a true pathogen if it is crab infection, and it also has multiple coexisting resistance pattern by which it acquires the resistance. It could be enzymatic or non-enzymatic or even mutation with the penicillin binding protein. And there are certain risk factors we have to consider for the development of crab infection. The first and foremost is the prior colonization with the MRSA has been an important risk factor for crab infection. Then prior beta-lactamase, especially carbapenem or fluoroquinolone use, bedridden patient, ICU admissions, central venous catheter, recent surgeries, mechanical ventilation, dialysis, malignancy, and steroids. So these are the risk factors for acquiring carbapenem-resistant Acinetobacter baumannii. So as we told already, there is no activity for ceftazidime avibactam or any latest antibiotic or combination for this organism. And ceftriaxone also has a disappointing result in the studies for the against the crab infection. So treatment of crab is actually a combination therapy. They advocate a combination therapy with at least 2 agents, ideally with in vitro activity proven. And the potential combination therapy could be any of these, but preferred one is ampicillin sulbactam. But it's not the usual dose. We have to give at least 24 grams in 24 hours. And for mild crab infection like UTI or skin soft tissue infection, we can use single agent and they advocate ampicillin sulbactam again in a higher dose other than the standard dose. And lastly, coming to the Pseudomonas aeruginosa infection with the difficult to treat resistance, the definition is it is not susceptible to the usual anti-pseudomonal antibiotics. And here also we can use ceftazidime avibactam, but the broad spectrum 5th generation cephalosporin, ceftalozine, tazobactam combination is actually preferred in this situation. And lastly, coming to the what are the preventive action, control, and intervention that you have to consider for this emerging issues. So there are healthcare environmental related commitment and intervention that you have to consider, and also community level programs. In the healthcare environment, we have to consider contact precaution of any patient who are infected or colonized with MDR bacteria. We have to have contact precaution. We have to keep him under the contact precaution till the patient get discharged, as per the WHO and American Society of Healthcare epidemiological report. And of course, hand hygiene, minimizing the use of invasive devices, cleaning and disinfection, proper cleaning and disinfection of the endoscopes, antimicrobial stewardship programs in all hospitals, and active surveillance of patients who are at risk of having MDR pathogen symptom, like routine rectal colonization screening. And in the community level, we have to reduce the over-the-counter availability of antibiotics. There should be proper legislation that it should reduce the use of broad-spectrum antibiotics from the livestock and fisheries, water body sanitation, clean drinking water, and proper sewage disposal management. So to conclude, antimicrobial resistance is a growing public health challenge at a global level. It can cause severe infection than other routine infection. It can cause severer infection, which can cause higher complication rate, mortality, morbidity, and ultimately treatment cost and hospital stay. So we have to improve and increase research and development area for the development of novel agent to safeguard the current and the future generations. There should be strict control on antibiotics prescription and use. And of course, it should be a multifaceted strategy we have to adopt, multi-sectoral resolution, not only the health industry, but also legislation, agriculture, and community level aspect. With that, I'm concluding the topic. Thank you for the patient listening.